So my last class was your introduction to the various bacterial strains that are used for cloning purposes. And at the same time, I also sensitized you that how these various bacterial strains, be it E. coli, be it other strains, they are being manipulated for better production of the protein. So they are manipulated in various terms in which, let's say, they are making changes that the protein gets folded properly, or you get an inducible expression of the protein, or let's say by promoting the transcript stability by making changes in these proteins, or ensuring that the codon bias does not affect the final protein production. And yeah, so all these points, these were frankly just some of the points that are being that I discussed with you yesterday. But otherwise, the bacterial strains are being engineered for better production of the final recombinant protein. Then I also told you that for cloning purposes in bacteria, plasmids and bacteriophages are two forms of vectors that can be used for transfer of the foreign DNA into the bacterial cell. And why we use plasmid, I told you. Is the slide visible?
Yes, ma'am.
Yes, ma'am.
Okay, okay. I heard some voice, so I thought that maybe there's some. Was there a question? Okay. So anyway, so yeah, so I introduced you to what plasmids are. I also introduced you to the fact that the size of plasmid is very important, because if the size is smaller, then you have the capacity to clone larger insert sizes. And also the introduction of the plasmid into the bacteria is comparatively much easier if the plasmid is smaller. Then I also introduced you to the fact that the copy number of plasmid, it varies between various bacteria, and also depending on which plasmid we are talking about. And there is a possibility to increase the copy number of the plasmid by certain ways, and one of which was introduction of chloramphenicol, by which you can increase the copy number of a particular, let's say, low or middle copy number plasmid to very high copy number by treatment with chloramphenicol. And this is where I ended my class. At the same time, I had also told you that there are various ways by which copy number is controlled in for a particular plasmid. And that's why, because now we have a basic understanding how the copy number is controlled, people have deleted certain genes to increase the copy number. And in that context, I told you about ROP gene as well as RNA1, etc. So now there are 3 small points that I just want to introduce you before I come to the point of how, what is the structure of plasmid vectors. So one is that every plasmid, it should have an origin of replication. If ORI is not there, then obviously the plasmid will not be able to replicate, and our whole purpose of having multiple copies of a cloned gene, it will not be possible. Then there is also a fact that ORI, depending on the ORI, it determines the host and the copy number of a particular plasmid. So I'm just rushing through these because these likely have been covered in the microbiology class. So you can please go through the notes again. Then there is something called as plasmid incompatibility, which I had mentioned in my last class also. And it is the inability of two plasmids to stay together in one bacterial cell. So if you have to introduce two different genes in a bacteria, and let's say they are present in not in one plasmid, but they are present in two different plasmids, then you definitely have to ensure that the two plasmids do not share the same ORI, because if they do share the same ORI or PAR regions, then likely they'll be highly incompatible. And why they will be incompatible? Because if they share the same ORI, then they will compete for the same machinery for replication. So in that context, it's better to look for the plasmids which have different origin of replication regions, and means which are definitely much more compatible as compared to if you choose plasmids which have the same ori. Then coming to the host range, I had a little bit mentioned in my last class also that there is some plasmids which have a very narrow host range, while there are some plasmids which have a broad host range. So the narrow host range, as the name suggests, these are those plasmids which can grow only in some bacterial strains, or maybe even one bacterial strain like that. So these are the narrow host range plasmids. The example is given here. And then there are certain plasmids which have a broad host range. And it means that these plasmids can exist and replicate in multiple bacterial species. So they have a very broad host range. And why do they have a broad host range? Because they have comparatively, or let's say they are less dependent on the bacterial host for their replication. So because they are comparatively less dependent on the host, so it is easier for them to exist in multiple host strains. So overall, all these points which I have mentioned with respect to plasmid, they have a bearing on the plasmid vectors. Size, I've already told you, smaller the size, larger is the capacity. Higher the copy number, you will have higher recombinant protein being produced. Although in those cases where the protein is toxic, you may prefer for the genes, for the plasmids which have low copy number. Host range comes into action if you want to introduce your recombinant plasmid into multiple hosts. So maybe in those cases you will look for a plasmid vector which has a broad host range. Otherwise, generally the ones that we have in the lab, and mostly we use bacteria only, then we go for this COLI1 based plasmids which have a narrow host range. Plasmid compatibility also is very, very important. When you have to introduce multiple plasmids for various purposes in the same bacteria, you will go for those plasmids which are compatible with each other. And if you do not take care of this particular point, you will ultimately lose one or the other plasmid over a period of time. Then last point is conjugative or non-conjugative. I've told you that for this biosafety purposes, generally the plasmids that we use, they are non-conjugative, or they have been made non-conjugative, so that whatever recombinant plasmid you have made and introduced into bacteria is not transferred to the other bacterial species and spread in the environment. So yes, all these points are important. And now coming to what are plasmid vectors. So plasmid vectors are the, let's say, the plasmids, naturally occurring plasmids, which have been manipulated to function as efficient vectors for cloning of DNA. So generally the range of DNA that can be cloned into plasmid is up to 10 kb. And that's why I mentioned that you will prefer a plasmid which is much smaller in size, but it does have all these features, so that you have the means you can clone much bigger insert sizes, because there is a limitation of the size when we talk about plasmid vectors. Now, what are the major features that are required for any plasmid to function as a vector? So one is small size, I've already told you. Then there should be a origin of replication. It's not shown in this figure, but without origin of replication, it cannot replicate and make more of its copies. So these are must. I mean, this is a must thing for a plasmid vector. Then to make a plasmid vector more efficient and giving it flexibility for cloning of various DNA fragments, in the modern vectors, they have introduced a multiple cloning site, which is nothing but a stretch of DNA in which multiple restriction enzyme sites have been introduced. Why these have been introduced? To give the user the convenience to clone his or her gene of interest in any of these sites, between any of these sites. Because if there was just one site available, let's say, for cloning purposes in the plasmid vector, then means we will be restricted that every time we should have, let's say, eco-R1 site on both the sides of our insert. So we don't want to be limited with just one side in which we might have problem in looking for directionality and all. So it's very, very useful that if you have a means a stretch of DNA in which multiple restriction enzyme sites have been already introduced in these commercial vectors. This is also, I want to bring to your notice that when we talk about the enzyme sites which are present in this multiple cloning site, then these enzyme sites, they are called unique restriction enzyme sites, because it is ensured by the person who has developed this plasmid vector that this enzyme site is only present in this stretch and not elsewhere in the plasmid. Because you can understand that, let's say if hint3 is present somewhere here, and if you digest your vector to clone your gene of interest, then obviously your vector will also get digested. So all these enzyme sites, they are unique in nature. Then so means but I would like to stress that MCS is not an essential feature of a plasmid vector, but yes, it is a feature which helps the final user in cloning multiple DNA fragments. Besides the plasmid vector, they generally have a selectable marker genes. And what are selectable marker genes? It means that if we want to identify a bacteria which has taken up a plasmid, then it will be great if we can find a mechanism that only those bacteria survive which have taken up the plasmid, while other bacteria, they die. It means we are looking for a selection for the bacteria which have taken up the plasmid. So most of these plasmid vectors, they will have these selectable marker genes, which usually are antibiotic resistance genes. So if we choose a bacteria which is not resistant to certain antibiotics, and we choose a plasmid which provides the resistance against that antibiotic. So obviously if we grow a bacteria in presence of that antibiotic, it will die. But the bacteria which has taken up this plasmid, it will survive because it has a resistance gene against that particular antibiotic. So all of these plasmid vectors will definitely have a selectable marker genes. Then there are certain other points that have been introduced in these plasmid vectors. So if any plasmid vectors just has ORI, MCS and selectable marker gene, we can definitely use it as a plasmid vector, or let's say cloning vector, in which whatever we clone here will make multiple copies because we have ORI in this plasmid. But we will not be able to, let's say if we want this particular fragment that we have cloned to be transcribed and translated, then we need certain extra features to be introduced into these plasmids. And what will be those extra features? They will be the promoter regions. So if this DNA insert is not preceded by a promoter region, then obviously it will not be transcribed, because then RNA promoter does not have any site to come and transcribe it. So if you want to make a expression vector, that is, you want your gene of interest to be transcribed and further translated, then you definitely need certain features. So what those features are, I will discuss those in a separate class on which will be totally focused on expression vectors. But for your information, the difference between cloning vector and expression vector is that expression vector is a subcategory of cloning vector in which the elements for transcription and translation of a cloned DNA fragment is there. Okay. Then in certain vectors, for the convenience of the user to sequence the cloned gene, is another feature has been added, in which they have given certain common sites which are present next to the MCS, or let's say they are on either side of MCS, that if any user wants to sequence the gene that has been inserted, there are some universal sequencing primers which are generally present in any company where you send your clone for sequencing. So generally those primers are T7 primer or M6 primers, and so many different types of primers are there which are called as universal primers, because the sites where they bind, they are present in generally all the plasmids that are being that are available commercially. So if I've cloned a gene, what I just have to do, I will send it to the company and I will say that please sequence it using T7 primer. So I don't have to design my own primer every time I generate a new clone. I will just say that please use your universal primers which are present in on either side of the MCS, and please sequence it from both the sides. Okay. Only problem could be that if your gene of interest is very big in size, then because one sequencing reaction at a time just gives, let's say, 500 or 600 base pair of reads, then you will not be able to completely sequence, let's say, a 5 kb insert just by having universal primers from either side. So in those cases, you will have to wait for the results of sequencing from either side, and then you will again design the primer from the ends of the sequence that you already know, and then go further and then go further and then go further. So probably this point will become further clear when I will take a class on DNA sequencing, the various strategies for sequencing a particular stretch of DNA. Okay, but in short, these are the various features that must be present in any plasmid vector. Okay, so this slide introduces you that in general, in various plasmids, especially bacterial plasmids that I'm talking about, which are the various antibiotic resistance genes, or let's say mostly they are resistant to which particular antibiotic. So generally they have ampicillin resistance or tetracycline resistance or chloramphenicol and kanamycin. These are, let's say, four very common antibiotic resistance genes that are present in most of the plasmid vectors. How these antibiotics kill the bacteria? So I've written all the points here. You can please go through it. But in general, what they do, either they, let's say, inhibit the synthesis of bacterial cell wall. But in general, what you will find that they prevent the protein synthesis by either binding to the 30S subunit or 50S subunit or by some other way. And finally, they prevent protein synthesis. So if protein synthesis is inhibited in a bacteria, then it will not be able to survive. So only when you provide a gene that can actually inactivate this antibiotic, or it prevents the entry of this antibiotic, then that bacteria can survive. So how those resistance genes are introduced into bacteria? With the help of plasmids. So what these resistance genes generally do, as I've written, you can please see, mostly they prevent the entry of the antibiotic into the bacteria, or in many cases what they do, they prevent the action of these antibiotics by preventing their binding to the 30S or 50S subunit, etc. And maybe there are many other antibiotic resistance genes which have different mechanism of antibiotic resistance. It could be that the drug enters, but then there is more of efflux, so it is not able to function at all. So please go through this slide just to have a brief idea that what are the various mechanisms for antibiotic resistance which are present in various plasmids. So coming to PBR322, what is PBR322? One of the very early vectors which was developed. So it was not a natural plasmid, but it was developed by bringing together the various elements to make a plasmid vector by various scientists. The names are given here. So let me introduce you to PBR322. So first let me tell you how the nomenclature of plasmids are. Generally, the small p is the first thing that you put before each plasmid name. It is generally in short means small case p. Then BR in this case refers to the scientists or the universities where this plasmid was developed. For example, in PBR, BR stands for Bolivar and Rodriguez, the two researchers who developed this. And for example, when I'll be telling you about PUC vector, so again, P is small and UC is big. And UC stands for University of California, that is the place where that vector was developed. So this is how the naming is done. Coming to the number is the fact that means in the lab, uh, the they give certain numbers, or maybe in the order that they were discovered and so on, or their screening purposes. And so let's say if PBR 322 was the early one, then they made some changes and then they developed 325, made further changes, they developed 327, 328, and so on. So this is how the nomenclature of any plasmid is done. So now coming to the properties of this cloning vector. What they have done is that they introduced, so let's say there was ampicillin gene, which was present in plasmid R1. And this is a naturally occurring plasmid. So ampicillin they have taken from here. TET R gene, that is the gene which provides resistance to tetracycline. It is derived from another plasmid R65, and another plasmid is there PBR320. The origin of replication in this particular plasmid, it is derived from PMB1, which is a close relative of a naturally occurring plasmid, CoLi1, which is that colicin producing plasmid. So basically what I want to tell here, that people engineer the plasmids, and still they are engineering new and new plasmids for based on the requirements of the user. So maybe means they have a whole series of plasmids. Maybe one has ampicillin resistance, some has tetracycline resistance, some has one ORI, some has other ORI, some has different MCS, some has different set of MCS, some has different promoter, and many other things which you'll understand when we'll go further in this whole course. So yes, yeah. So one fact is this, that how these three things were brought together in one plasmid. Second thing is, you will notice that it doesn't have a multiple cloning site because it was one of the very early ones. And in the improvements of this vector, the MCS was introduced by the scientists later. So in this, how cloning was done and how selection was done is what I want to introduce you to. So here you know that there are two different antibiotic resistance genes, and each of these antibiotic resistance genes, they have some or the other restriction enzyme site. So now let's say that I use BAM-H1 for cloning my gene of interest. Okay, so now I have my gene of interest. I cloned it into PBR322 by ligating it, means first restriction digesting it by BAMH1 and then performing a ligation reaction. And then whatever the ligated products are, I introduce those into bacteria. So once I introduce those into bacteria, I have two options, means three options that take place. So one in which just bacteria is there and I grow it in presence of ampicillin or tetracycline, that bacteria will die because it doesn't have a resistance gene. The bacteria which takes up the original PBR322 where ligation has not yet taken place, it will survive both under ampicillin as well as tetracycline because both of these resistance genes, they are intact. Now consider a situation in which the cloning has taken place and I have a recombinant plasmid where inside this BAMH1 I have my gene of interest. So what will happen, that because my gene of interest is cloned here, tetracycline resistance gene, it will be interrupted, and because it will be interrupted, what my bacteria, how it will behave, it will be resistant to ampicillin, but it will die in presence of tetracycline. So I think this is likely clear that it will survive if it takes up intact PBR322. It will die if it does not take up plasmid, and it will survive in presence of ampicillin but die in presence of tetracycline in case it takes up the recombinant clone. So how do you identify the bacteria which has the recombinant clone? Because ultimately that is the one which is dying. So they used to do a technique called replica plating. Okay. So what is replica plating and why am I discussing this when in current situations we do have the option of having multiple cloning site, and we have various screening markers also, which makes it much easier to identify the bacteria which has taken up the recombinant plasmid, because replica plating is one technique which is also currently being used for various other purposes as well. So coming to what replica plating is. So here is my master plate in which I have ampicillin resistance. Okay. So all the bacteria here, whether they are transformed, uh, just a minute, here what example they have taken. Okay, so here also they have cloned in tetracycline only. And my point is that first you grow a master plate in ampicillin. To my understanding, everything will grow here except the bacteria in which it has not taken up the plasmid. Then you transfer these bacterial colonies with the help of a velveteen pad or let's say some membrane, and you take it up on that particular membrane. Once that membrane has taken up these replica of these bacterial colonies, you put those, you let's say stamp those membrane in a tetracycline containing media. So here what will happen, again I have told you that here the tetracycline cells will grow, and those bacteria will grow which have taken up only the non-recombinant plasmid. So those only will grow. So then what I can do is I can compare this plate, my original plate, with this plate, and I can identify the bacteria which are not present here. Okay, means this and this are not present here. It means these ones in my original plate were the ones which are the recombinant cells, that is the bacteria which has taken up the recombinant plasmid. So then I can, pick up these bacteria and I can grow them in presence of ampicillin, and then I can isolate my plasmid, and this is how I can amplify my recombinant clone into multiple copies. I hope this is clear. If not, please let me know. Okay. Okay, so then further improvements were made with respect to PBR322. What they did was that they realized that, see, you want to make your vector comparatively small in size for the reason of having larger insert. So they identified a region of around 1000 base pair, which was probably not required for PBR322 to function as a plasmid vector, because the ampicillin and TET genes were intact, the ORI was intact, and many other segments which are important for it to function as a vector, they were intact. So this region was deleted. So one improvement was this, that it is shorter than PBR322, but at the same time it was realized that for whatever reason, when they removed this segment, the copy number, which in PBR 322 was around 15 to top 20 copies, it was increased to 30 to 45 copy number per cell for PBR 327. So these were the two improvements, and apart from that, They also realized that this particular segment, it led to the loss of conjugation ability. So, which I told you is very important for biological containment. So, these were the improvements. If I ask you what are the improvements of 327 over 322, it is shorter size, higher copy number, and the loss of conjugation ability. Then came a major advance as compared to PBR 322 or 327. These are called as the PUC vectors, and various PUC 18 or PUC 19 vectors are there. And what improvements it had was, one, that here the multiple cloning site was introduced. I just come back to how does it look like and all. But yes, one improvement was MCS was introduced, and another improvement was that now we didn't want to go through this whole set of replica plating and then identify the recombinant bacteria, but rather here a screening marker was added, and in this case it is the LAGSI, or let's say blue white screening, that was introduced here. So what that screening methodology is and what is the idea behind it, I'll just come to that. But yes, these were the two major things, MCS and the presence of this screening marker. And one more thing was that in PUC vectors, when they were actually manipulating these vectors, then a chance mutation took place in its ORI region, and the copy number was drastically increased. So the PUC vectors, it had around 500 to 700 copies per cell due to a chance mutation in the ORI. So now you can understand the importance of ORI and how still various scientists, they are trying to manipulate the ORI in such a way that the copy number is increased for any particular plasmid that they are working on. Okay, so now coming to first the MCS. So MCS, in this case, it was around 60 nucleotide long DNA region, which had site for 13 different restriction enzymes, which is definitely a very, very big advantage when you talk in terms of practicality when you are cloning your gene of interest. So the advantages are you have more options. Second, you can maintain the directionality, because if let's say in the case in PBR 322, you just had, let's say, BAMH1 site. So you cannot go for directional cloning because only one site is there. But here you can definitely clone, let's say, in SMA and PST1 or XBA and SAL1, and so many options are there. So you're not limited as a user. Then if I want to excise out my DNA with the same sites or maybe with different ends, even that is a possibility. Let's say I clone my DNA fragment in BAMH1 and PST1, but now I want to clone it in a vector which has, let's say, SAC1 and HINT3.1. So what I can do is I can cleave my DNA fragment out by SAC and HINT3.1 and clone it in another vector. So the excision, it can also be by the same, let's say I've cloned in BAM and PST1. I can also excise it using BAM and PST1 and subclone it in another vector of choice. And another option is you can use different enzyme sites also. That option is also there. And if you want to do restriction mapping, even that you can do by the various enzyme sites which are present on the either side. And if you want to know the directionality of a clone, even that option is there, which we had discussed previously also. Let's say you have cloned, you had no option, but you could clone only it in XBA1. Let's say your DNA fragment is present in XBA1 site, and now you want to know whether your DNA fragment has gone in the right direction or not. So you can utilize the various enzyme sites which are present in this DNA fragment, your DNA fragment, and any of the enzyme site which are present close to the this cloning site. So then you can know, for example, you cloned a fragment of 500 base pair, and towards the end of the 500 base pair, let's say you had a SMA1 site. So then if I digest my recombinant clone with SMA1, then if it is in right direction, I will get a 500 base pair around fragment. But if it has been cloned in the opposite direction, then hardly it will be, let's say, 20-30 base pair fragment that will come out. So this we had discussed previously also, but here with respect to MCS and its advantages, I'm just revisiting that. So this is, so means various names have been given. It's called polylinker, it's called unique restriction enzyme site region, or it is also called as multiple cloning site region. Now coming to the screening ability, means is there some way where you can identify the bacteria which has taken only the recombinant plasmid but not the original plasmid? So in that context, there is a way to do it, and one of the ways blue-white screening. So in blue-white screening, they have utilized one enzyme called beta-galactosidase coded by LAGZ. So this beta-galactosidase, when it acts upon a substrate called X-gal, it gives a blue color. Okay, one fact is this. Second fact is that beta-galactosidase, it acts as a homotetramer. So only when these four monomers, they come together, it becomes active. It is only then it is active in nature. So these four fragments, they are brought together by a alpha peptide, around 1 to 63 amino acids is that region. So when this alpha peptide is there, it brings these four monomers together, and once they are together, they are, and if X-gal is given, then it gives rise to blue color. Okay. So what they have done is that it is a basic observation that how beta-galactosidase work, and look at its application. So what they did was that in the plasmid, they cloned they have this alpha region. You remember this alpha peptide region? So alpha peptide region is carried in the plasmid, and bacteria contains rest of the whole beta galactosidase, which lacks the N-terminal region. Okay, so in a way they have divided the whole polypeptide of LAGZ into two different fragments. One is this mutant allele which lacks the initial N-terminal domain, while the N-terminal domain which codes for alpha peptide, it is present in the plasmid. So what they have also done is the fact that in the plasmid where alpha peptide region is there, in middle of that they have added the MCS site. So now what will happen that let's say a plasmid in which my gene has not been cloned, what will happen, this alpha peptide will be coded here, and in my bacterial strain, because rest of the polypeptide was there, it will code for those four monomers. So because those four monomers are there, then this alpha peptide will go and bind to those four monomers, will make it a complete homotetramer, which is functional. And when I introduce into this bacteria the X-gal thing, then it will give me a blue color. This is for non-recombinant plasmid. Now, what will be the scenario if I have a recombinant plasmid? My gene of interest will get cloned here in the restriction enzyme site, and because of which my alpha peptide, it will be interrupted. If my alpha peptide is interrupted, what will happen that rest of the protein will get encoded by the bacterial strain, but it will remain non-functional because the monomers remain separate. They are not brought together by alpha peptide. So what will happen that even if I introduce this bacteria, I treat it with X-gal, I expose it to X-gal, it will not be able to metabolize it. If it is not be able to metabolize it, then we will get only white color bacterial colonies. So now what's the scenario? If I transform this puck vector after ligation into bacteria, I still have three different scenarios. One bacteria which does not take up the plasmid, that will die in a plate, in a LV agar plate where I have put ampicillin. So my bacteria which has not taken up the plasmid, it will anyway not survive. The two types of bacteria that will survive will be those bacteria, one which has taken up the non-recombinant plasmid and one which has taken up the recombinant plasmid. So the one which has taken up the non-recombinant plasmid, it will give a blue color because alpha peptide was intact. While the bacteria which takes up the non-recombinant plasmid, it will give a white color because it is unable to make functional alpha peptide and functional beta galactosidase. Okay, so this is how your this plate will look like. And this LB agar plate, for your information, it contains ampicillin, and it is means spread, exgal is spread on this particular plate. So as a result, bacteria will get a chance to have a substrate exgal to act upon, and if it has intact beta galactosidase, it will give blue color. But at the same time, just because you got a white colony, please don't be happy that for sure your gene of interest has been cloned, because it could be that even if some other DNA as a contaminant, let's say in your ligation mixture, it is present there, and even if that gets ligated in this MCS region, even that will interrupt the alpha peptide, and as a result you will get a false white positive. So it is important that if you get the white colony, you screen 3-4 white colonies, you look for your gene of interest for the same size, and once you get that, okay, my gene of interest means same fragment is coming out when I'm digesting my plasmid with a restriction enzyme in which I had cloned, then you should again not be completely happy unless you sequence it, because there is a chance when you're doing your PCR that you can introduce some mutation in your gene of interest, so which can only be figured out once you clone it and you send it for sequencing. So once you, after sequencing, if you find that okay, there is a 100% match between your gene of interest and the means the sequence that is available on the web, then only you should be completely happy. So again, coming to these Puck vectors, their nomenclature, they were developed at University of California. We have different series, 8, 9, 18, 19, the details of which are given in one link that I've given in the notes section. But in general, because of some change in the ORI as compared to PBR 322 or 327, it is present in extremely high copies, a very big advantage in many scenarios. There is an option to look for recombinants by blue-white screening, which is much easier method as compared to, if you remember that replica plating, it was much cumbersome. And of course, the presence of MCS site, which provides various flexibilities for the user, and also the option for subcloning in other set of vectors which are important for various purposes. Okay, so what the new, let's say, commercial vectors that are available, what they generally do is that they keep MCS site common between different types of vectors. So what happens that if I want to move my gene of interest from, let's say, this Puck vector to, let's say, MP series vector, then it is very easy. I will cut with SMA and PhD, let's say my gene is cloned here, and I will cut my MP series vector with the same enzymes, because in this vector also they are present in the same orientation. So I will cut my recombinant plasmid. The DNA insert, it will be it will be generated, and you can purify that DNA insert and then just clone in your other vector. So you don't have to again go for amplification by PCR, introduction of restriction enzyme sites on either sides of the primers, and then cloning, etc. So subcloning becomes much easier with the help of MCS. So the last vector that I want to discuss today is PGM3Z vector. So in this vector, they have made it in a way, the option has been given as compared to PUC vectors. They have introduced promoters on either side. So they definitely have the same ORI is there, the ampicillin resistance is there, the LAGZ is there, and the multiple cloning site is there. But the two changes that they have added here is the presence of promoter region on either side. Now, if you notice the promoter region, they are derived from bacteriophages. So this T7 promoter, it is not a native of bacteria genome. It is present in the T7 bacteriophage, and sp6 promoter has been derived from sp6 bacteriophage. So why they have chosen these promoters? I had told you in my previous class also that T7, these bacteriophages, when they undergo lytic life cycle, they have to very quickly make their proteins, etc. So they have very efficient promoters as compared to most of the bacterial promoters that we are aware of. So because they are extremely efficient and give rise to a large number of transcripts at a given time, so that's why these promoters are preferred as compared to any bacterial promoters. So it is for high expression. Now, one question could be that, okay, let's say clone my gene of interest here, and promoter is here, so I will get a transcript which is specific to my gene of interest. So what is the need to add sp6 promoter on the other side of the gene of interest? So that is added because in many cases there is a possibility that maybe I'm not interested in just the sense RNA. I am also interested in the antisense RNA. Maybe I want to make an antisense probe against a particular transcript while performing, let's say, in vitro transcription. So in those cases, it is good that I have option to generate a transcript from the other side also, which will generate the antisense RNA against a particular transcript that you are interested in. So this is a general introduction to the various plasmid vectors. So at the end of these yesterday and today's lecture, you should be aware of the fact that what are bacterial strains that are generally used, what changes you can make in bacterial strain to make them the best host vectors, and also what are the properties that are essential for any plasmid to function as a vector, and what changes have been made in various regions in the plasmid vector to make them very efficient in nature and for a particular purpose that a user is interested in. You can make changes in ORI, you can introduce various antibiotics, you can have different types of screening markers. Is blue-white screening the best screening? Maybe it is user friendly, but maybe it's not the best screening. It does have an option to give false positive. Maybe you want to go for different types of screening strategies, etc. So I'll revisit the screening strategies and maybe more selectable markers in my subsequent lectures. But I just want to introduce you to the fact that when you're working in different labs which are working on different organisms, you have plant, some labs are working in plants, some in mammals, some in bacteria, which are not limited to E. coli. For example, Dr. Preeti's lab, it's working on Gordonia bacteria, and maybe so many different bacteria, the names I don't remember. So maybe in their context, the requirement for a plasmid vector is different. And they are looking for different type of ORI, which will be, more efficiently working there, or they are looking for the incompatibility and a partition locus, etc. So many different points are there, which at least you should be aware of. That's my point. Okay, so that's all from my side today, and I
Ma'am, I
am
have one question.
open for questions. Yes.
Ma'am, in the last slide, which has both T7 and SP6 promoters, how do we selectively activate one of these?
By providing the T7 RNA polymerase, which is specific to it. So likely, see, if you remember my previous class where I introduced that what changes have been made in the bacterial strains to make a particular host strain means let's say a particular gene of interest inducible in nature. So in that context, I told you that in some plasmid vectors, they have introduced T7 promoter. But because the bacteria itself does not have the T7 RNA polymerase, it just has E. coli RNA polymerase. So what they have done, they have introduced into the E. coli the RNA polymerase for T7, which is called T7 RNA polymerase. So, and in the same bacteria, let's say I introduce this plasmid in a particular bacterial strain. So in the same bacterial strain, there will also be a promoter against, SP6 RNA polymerase gene present in the bacterial strain. So what they have done, they have made those polymerases inducible in presence of certain signals. So it could be that you make the T7 RNA polymerase inducible in presence of IPTG, and you make sp6 RNA polymerase inducible in presence of, like say, rhamnose, or let's say some other, let's say tryptophan inducible. So I can then regulate that, okay, in presence of IPTG, only my T7 RNA polymerase will get activated. It will come and bind to the T7 promoter and generate the transcript in this direction. However, if I want my antisense transcript, I will not give IPTG treatment. I will give the other treatment to ensure that only sp6 RNA polymerase is generated, and it goes and binds here and generates the RNA in the opposite direction.
Okay, ma'am. Thank you.
Okay. And this T7 RNA polymerase and T7 promoter thing, I'll revisit when I'll be telling you about PET vectors, which are expression vectors. And in most of the labs which are working on protein synthesis for sure, where you want to purify protein of interest. So in those contexts, PET vectors you should definitely be aware of, and they have this system. So I'll revisit this for maybe better explanation. Okay, any more questions?